Welcome everyone to today's keynote lecture. So this year we're really thrilled to have Dr. Jeff Biernaskie from the University of Calgary, who will be presenting the Bernard Langer Lecture in Health Sciences. Dr. Biernaskie earned his PhD in Neuroscience at Memorial University, studying the effects of focused rehabilitative experience on brain plasticity following stroke. He completed postdoctoral training studying skin and neural stem cell biology at the Hospital for Sick Children right here in Toronto. And in 2009, he joined the Faculty of Veterinary Medicine at the University of Calgary, where he's currently a professor in stem cell biology and regenerative medicine. He's a member of the Alberta Children's Hospital Research Institute and the Hotchkiss Brain Institute, and he holds the Calgary Firefighters Burn Treatment Society Chair in Skin Regeneration and Wound Healing. So before we get started, I would just like to remind people that throughout the talk, you can look to your right of the screen and go to the session's Q&A tab and just input any questions you might have. You can also vote on other people's questions using the thumbs up feature. And then at the end, I will ask some of the highest voted questions on behalf of the group. And so now, without any further delays, I'd like to welcome Dr. Jeff Biernaskie, who will be presenting his talk on deciphering the cellular and transcriptional recipe enabling skin regeneration.
Okay, can everyone see my slides and, whoops. Can everyone see my slides now and hear me okay?
Yes, it looks good.
Okay, I'm just going to advance. Okay, so thank you very much for that very kind introduction. I want to start by thanking the organizing committee, particularly Drs. Narang and Zai, and all of the graduate students and that have contributed to organizing this fantastic IMS Research Day. I really enjoyed the poster presentations yesterday, and the talks this morning have been really excellent. So thank you very much for the invitation. I also want to just tell you how thrilled I am to give the Bernard Langer Lecture, particularly because Dr. Langer is regarded as such a visionary in surgical medicine, and based on his appreciation for the importance of research and clinical training, he pioneered a world-class surgeon scientist training program here in Toronto. And so, you know, and actually, I work with many of the graduates of that program here in Toronto or here in Calgary, and so it's really an incredible honor to be invited to tell you about our work. But at the same time, you know, entirely humbling to be mentioned alongside of someone who has accomplished so much. So I just wanted to acknowledge Dr. Langer's incredible contributions to medicine in Canada. And for inspiring researchers and mentors like me to try to do better. So today, what I want to tell you about is our interest in scar. And so my lab has a variety of different interests, but you know, a primary focus of our work is really in scar. And you know, that interest for me really started as a result of our interactions with our clinical collaborators in the Calgary Firefighters Burn Treatment Center, namely Dr. Vince Gabriel and Dr. Duncan Nickerson. And this is, you know, and through those interactions, I had the opportunity myself and my students to engage with various patients within the unit. And you know, it was examples like this one, where this is a woman that came into the burn unit, not actually from a burn, but from necrotizing fasciitis. But you can see that the extensive scarring that she has on her neck and shoulder region, and this is after 7 different surgical procedures to repair these skin wounds. And I think what really impacted me was what I life that scar can actually have. So for example, with her, I mean, you can see that you can see the distortion of her mouth which really impacts her ability to speak and to eat and drink. She has immobility in her shoulder, and you can see there's aesthetic issues as well. And so there's lifelong impairments in mobility, itch, pain, as well as psychological impacts due to this scar. And so this was really, one of the reasons that we in my lab were inspired to try to understand the pathogenesis of scar and how we might be able to use that knowledge to develop treatments to prevent scar from happening and to promote more regenerative healing. And so for those of you that don't work on skin, this is just a schematic of a cross-section of what the skin looks like. And so this upper layer is called the epidermis. It's really the outer covering of the skin. And then beneath that is this specialized layer of connective tissue called the dermis. And within the dermis are a variety of different appendages like hair follicles, sebaceous glands, sweat glands, and a network of both vascular supply and sensory innervation throughout that connective tissue. But it's really those fibroblasts and within this dermal layer that I'm going to focus my talk on. And the reason for that is because it's those fibroblasts that are the cells that are the most important players in formation of scar during the wound healing process. And so, the question that we're most interested in is really what drives dermal fibroblasts to become scar forming rather than becoming regenerative. So this is a schematic of what typically happens following a deep skin injury that affects the dermis in adult mammals. So the default response is an activation of surrounding fibroblasts that then migrate rapidly into a lesion. They form this sort of disorganized mass of cells within the lesion itself, and then subsequently begin to secrete excessive amounts of extracellular matrix. And it's that disorganization and production of extracellular matrix that basically forms a non-functional tissue that fills in that defect. And so the goal is really to try to overcome that process and to divert those fibroblasts into permissive, to generate a permissive environment within the skin in order to enable complete regeneration and restoration of appendage formation, so generation of new hair follicles, new glands, restoration of fat depots within the skin, and eventually a more appropriate sensory innervation within that healed tissue. And so the way we go about studying skin regeneration is by using various mouse models, and then in the second half of the talk I'll tell you about an alternative model that we're using to study this process. But in the first set of experiments, I'll tell you about these wound experiments that we've done in mice. And so this work was pioneered actually by Mayumi Ito and George Katsarelis at University of Pennsylvania. And what they did was to demonstrate that in mice, as well as in rabbits, which had been demonstrated almost 3 decades earlier, is that if you make a small wound, a full thickness wound through the skin of a mouse, you basically form a fibrotic scar, typical of what you might see in other mammals. So you get reepithelialization, this is the epidermis here, but you get a filling in of that wound with sort of linear organized fibroblasts, and an absence of any of those appendages that I told you about before. So no hair follicles, no glands, no adipose. So it's basically just a fibrotic scar. But interestingly, if you change the size of that wound, so you make that wound much larger. So in the small wounds, these are about 8 millimeters in diameter. But in a large wound, when you increase it, the size of the wound to about 1.5 or to 2 centimeters in diameter, you start to see a process that is not only fibrotic, but also contains regions of at least some regeneration. And so this is a top-down view of one of these lesions. And you can see that the outer region of this wound is fibrotic, but in the center, you can see these blue stained structures here. And these are actually newly formed hair follicles. And this is another example of one of these lesions as well. And so what's happening is the blue staining is actually for alkaline phosphatase, which stains the inductive mesenchyme within hair follicles called the dermal papilla. And so this is a cross section of one of those, of the center of one of those wounds. And this is one of these newly forming or neogenic hair follicles within the center of the wound. And so what this really told us was that under some circumstances in mammals, there are instances where we can actually perform at least a modest level of regeneration. And it also told us that there are regionally distinct or regional distinctions within a wound that are permissive for regeneration, and then other regions, like in the periphery of the wound, that continue to form scar. And so what we wondered was, what's different about fibroblasts in this regenerative zone versus fibroblasts in this fibrotic zone that enables them to become regenerative? So what are the molecular changes that occur that underlie this regenerative competence? So just specifically, what we wanted to ask was, what's the source of these regenerative fibroblasts, and more specifically, are they derived from hair follicle associated fibroblasts, or potentially from extrafollicular or interfollicular dermal fibroblasts? And then, like I said, what are the transcriptional differences underlying adoption of regenerative or fibrotic fibroblast fates? And I'll just mention that the work that I'm going to tell you about has been done entirely by Dr. Sepide Abbasi, Sartak Sinha, and Elodie Labi, 2 or 3 very talented trainees in my lab. And so the way we started to approach this problem was we basically made large wounds or small wounds on the back of mice, and then profiled. Well, basically, we performed lineage tracing or fate mapping experiments to mark populations of fibroblasts in order to discern the origins of the fibroblasts that are participating in this wound healing response. And then we dissected out fibroblasts from either these peripheral scar-forming regions or from the central regenerative regions in order to understand those molecular changes that may underlie these different responses to injury. And then we profiled them based on single cell gene expression, as well as ATAC sequencing, and then verified some of those changes that we observed with RNA scope staining within the skin. And so I should backtrack a little bit and tell you one of the reasons why we thought maybe the source of these regenerative cells might be coming from hair follicles is because years previous we had identified a mesenchymal progenitor cell that resides within hair follicles, at the base of hair follicles. And the function of those cells is to really proliferate and to generate new fibroblasts in order to sustain these mesenchymal populations at the base of every follicle that are really critical for instructing epithelial cells during hair follicle regeneration. And if you don't know, the hair follicle is really a fascinating sort of mini organ, in that it's really one of the only structures in mammals that continue to regenerate throughout our lives. So it undergoes this process of degeneration and regeneration, and relies on this really intricate on two sort of populations, the mesenchymal population and a keratinocyte population, and this intricate communication between those two populations that allow this process to happen. But this population that I'm referring to, that we call hair follicle dermal stem cells or progenitor cells, this is one of these cells that we've labeled here in green. What's unique about them is that at the onset of this regenerative process, you can see that they reside just on the outside of this inducted dermal papilla structure. At the onset of anagen, it's these cells that are activated to start dividing. And when they do that, as the follicles go into this sort of anagen or growth stage, you see an expansion of those cells, of those progenitor cells. So they typically reside down at the bottom of the follicle, and they give rise to new cells that will then populate this inductive dermal papilla structure, but also give rise to this connective tissue sheath that wraps around each of the follicles, and basically provides a niche for the hair follicle, and a niche that allows continuous communication between the hair follicle mesenchyme and the epithelial stem cells that reside within the bulge region of the follicle. And so we wondered whether potentially this progenitor population might be activated and maybe recruited into skin wounds in order to participate, particularly in these new hair follicles that are being generated within wounds. And so what we did was to first use this transgenic fate mapping approach that we had used before, and that's using an alpha smooth muscle actin CRE-ERT2 promoter that's crossed to a YFP reporter. And so when you do this, when you apply tamoxifen in the uninjured state, what it does is it marks the dermal progenitor population at the base of the follicle, and then all of their progeny within the hair follicle. And what's important to note is this does not mark dermal fibroblasts in the resting state of the skin. Okay, so it only marks this progenitor pool within the follicle, along with some smooth muscle cells within the vessels. But here's a cross-section of one of these large wounds. And so we've labeled the cell, the dermal progenitor population within follicles within the skin. We wait 3 weeks and then sort of for a washout period, and then we give a large wound, and then we look to see whether there's any contribution of those green cells from these hair follicles within the large wound itself. And so here's a blow up of one of the central region of one of these wounds, and you can see these neogenic or newly formed hair follicles that arise within the center of one of these large wounds. This is a close-up of one of these follicles. The red is standing for keratin 14, these keratinocytes, and then green is showing the YFP labeled cells that were previously residing within the hair follicles. And you can see that a small number of these dermal progenitor progeny have migrated away from pre-existing hair follicles into the wounds, and actually a very small number of them have actually participated in formation of these new hair follicles. So what that tells us is that's one small source of cells that may be participating in this regenerative response. But it also tells us that there has to be another source of fibroblasts, and the majority of this regenerative response actually does not come from pre-existing follicles or any cells associated with the follicle, but actually must come from fibroblasts within the interfollicular regions of the dermis. And so in order to capture those cells, what we did was to use a different reporter, and this is called HIC1. This is based on a gene called HIC1, or hypermethylated in cancer 1. And this is a gene that we had found was expressed in dermal progenitors within the hair follicle, but also is expressed in fibroblast progenitor cells that reside within the deeper layers of the dermis. And this is in collaboration with Dr. Michael Underhill, who also identified this marker, or identified HIC1 as a marker of mesenchymal progenitors in a variety of other organs, particularly in the heart and in skeletal muscle, giving rise to both fibroblasts and adipocytes. And so we use this HIC1 CRE-ERT2 strategy in order to mark not only fibroblast progenitors in the hair follicle, but also fibroblast progenitors in this interfollicular dermis. And with the goal of labeling all of these sort of wound responsive fibroblasts that would be moving into the wounds during this reparative process. And so again, we labeled these cells with tamoxifen, waited 3 weeks for a washout, and then gave one of these large wounds or small wounds, and then looked at the fate of these cells. And you can see right off that this HIC1 lineage that we're marking labels the vast majority of cells that are moving into the wounds during this reparative process. So you can see the extent of red cells throughout the injured dermis. It does not mark any of the epidermal layer, which you can appreciate here, either in the epidermis or in the hair follicles. But what's interesting and what was really remarkable to see was that this HIC1 lineage is actually the primary contributor to all of the cells that are giving rise to these new hair follicles. So all of these inductive fibroblasts that are moving in and initiating this regeneration of new hair follicles, as well as regeneration of new fat within the deeper layers, which comes a few weeks after this, what I'm showing you here. All of these cells are coming from this HIC1 lineage of fibroblasts. And so what we wanted to know was now that we'd been able to label the source of these cells, and to determine that these are coming from largely from non-follicular sources, we could now try to understand the transcriptional differences between those fibroblasts that are moving into the peripheral regions that become fibrotic versus those cells that move into the central regions and actually engage these regenerative processes. So what we did was to basically sort out all of these TD tomato positive cells, and then subject them to single cell RNA sequencing analysis using this 10x genomics platform. We also compared these to not only just the large wound scenario, but also to the small wound scenario in order to get a purely fibrotic fibroblast response. And so this is now single cell RNA sequencing data. So this is a t-SNE plot. If you're not familiar with this type of data, what this is showing you is thousands of cells that have been isolated and plotted in two-dimensional space based on the ensemble of their transcriptional profile. And so cells that are closer together have overlapping or similar gene expression profiles. And so the first thing that I'll show you is this is in purple. These are uninjured fibroblasts, and then you can see that in the small wounds at 8 days after injury, you can see that the transcriptional profile now shifts following that injury. Again, it shifts even further when you look at the at 14 days, so they move to this green population. But what I want you to make note of is the difference when you make a large wound, and that you have, and that those large wound fibroblasts are shown in red here. And you can see that there is certainly overlap between the small wound and the large wound, but there are certainly some very distinct populations of cells within this large wound, which is indicative of these divergent responses. And you can better visualize that here when you just look at a large wound, and you discriminate those by their position within the wound. So the blue cells are all from the periphery of the wound, and then the red cells are those that have been isolated from the center of the wound. And when you plot them using the t-SNE space, you can see that although there is a lot of overlap in here, you can see that there are certainly a shift in the transcriptional or in gene expression profiling between the cells in the center compared to those cells in the periphery. And so what we started to do was to look at some of these profiles to understand what genes are being activated or what genes might be suppressed in these different populations that are either regenerative or scar forming. And so we've done this by looking at either the periphery or the center, or the center or the upper versus the lower. And so the upper, much like the upper part of the center is regenerative, the lower part is fibrotic, very similar to what you see in the periphery of a wound. And so when we compare these regenerative zone, or when we compare scar forming to regenerative, we see activation of known fibrosis-related genes being enriched within these fibrotic regions. So genes like DLK1, SCA1, and MEST, all of which are known to be pro-fibrotic. But when we look at these central regenerative regions, we see activation of genes that are typically reserved for embryonic skin development. So things like CRABBP1 or FABP5 or PRSS35 all were highly enriched within only or largely within the central regenerative zone, and many of these are expressed within the inductive fibroblasts during very early stages of skin development. And so it looks like what's happening is that these regenerative zones are actually co-opting regenerative programs in order to recapitulate development in order to regenerate the skin. And so these are images of some of those genes here showing the distribution of gene expression across one of these large wounds. And so this is the full extent of one of these large wounds, and this is a cross section through the skin. So this would be the epidermis up here, this sort of black layer. But what you can see is those two regenerative genes, CRABBP1 and PRSS35, are both enriched within this upper region as opposed to the lower region. But you also see an enrichment in the central region. So here this is where you've got these new hair follicles that are being formed. This is also the site where you'll see later adipogenesis. But you can see that there's an enrichment for those transcripts within this region, and this starts to diminish as you move to the peripheral regions of the wound, and certainly as you go deeper into the wound. We also observed genes that are like SOX18 that are really absent in postnatal skin and really only active during early development. And you can see in these neogenic follicles, so these newly formed follicles within the wound, they're re-engaging genes like SOX18 in order to recreate these new follicles. So the next thing that we did was to look at, is to further examine this transcriptional and changes that occur in order to drive the functional divergence between these different populations of fibroblasts. And so one of the things that we did was to look at, or to use a program called Scenic, which allows us to look at downstream network activity from of various transcription factors. And so this gives you sort of a functional readout of those transcriptional factors that are truly active. And what's interesting is that what we found was a discrepancy between transcript and the activation of those downstream networks. So if we look at the UMAP plot here, what we see is the fibrotic cells are typically shown, the fibrotic cells in the periphery are clustered down in the bottom of this plot, whereas the regenerative cells are in the upper part, just to orient you. But when we look at the transcripts for genes like CD59 or PRRX1, we see that they're pretty uniformly distributed across both populations, particularly PRXX1. But when we look at the actual downstream activity of those for those that are associated with those transcription factors, we see a discrete difference in activity. And so here you can see that both of these networks are highly active in the fibrotic fibroblasts and seem to be repressed in the regenerative ones. Similarly, if you look at genes that there's also genes that are or transcription factor networks that are highly associated with this regenerative response. So THAP11, GTF3A, and RAR alpha all again are uniformly distributed at the transcript level, but we see very discrete activation of those downstream networks in the regenerative zone. And so what this suggests is that there may be some post-transcriptional modulation that's occurring in order to drive this functional divergence within these fibroblasts. And so to probe this a bit further, we looked at potential epigenetic changes that might mediate this functional divergence. And so we applied single cell ATAC sequencing to look at this. And so as an example, here you can see now 4 different regenerative genes. So RAR alpha, beta, gamma, as well as RUNX1, where you see uniform distribution of transcript across the majority of cells, both fibrotic and regenerative. But again, we see discrete network activity only in the regenerative populations of the cells. And then when we looked at our ATAC data to look at accessibility for transcription factor binding motifs that are specifically associated with those genes, we see that there is a motif enrichment for those genes. So as an example, here's for motifs for RUNX1 and for RXR alpha, both of which are highly enriched, suggesting that that what we see is greater accessibility within the chromatin to those particular genes that's being enabled within that regenerative population of cells. And so it looks like it's not only transcriptional changes that are occurring, but also epigenetic changes that are also underlying this acquisition of regenerative competence. So the next thing that we did was to begin to perturb these mesenchymal cell regulators that we'd identified during skin regeneration. And so we began to apply different drugs, either agonists or antagonists, to skin wounds to see if that could influence the regenerative response. And so this is basically making large wounds on these animals and then counting the number of neogenic hair follicles within each wound. And so in a control animal or a vehicle treated animal, you see on average around 10 new follicles within each wound. If we apply a retinoic acid antagonist to that wound, we we see a significant reduction in the number of follicles that are generated. Similarly, if we block RUNX signal, also reduce the number of follicles. But if we promote retinoic acid signaling by application of an agonist, we can see that we can then increase the number of neogenic follicles within that wound. So this really suggests that these various pathways are really making a functional contribution to this regenerative competence. The other thing we did was to go back to this HIC1 lineage. As I told you, that HIC1 is a transcriptional repressor, and what it basically does is act as a brake on proliferation. And so what we did was to ask, if we took the brake off and allowed these fibroblasts to proliferate at a greater extent, would that change the outcome of regeneration? And so we performed wounds in alpha-synuclein Cre-ERT2 mice in which they had HIC1 flox allele, and that allowed us to basically delete HIC1 following injury. And so this was done during the week following the injury, in order to allow these wound responsive fibroblasts to increase their proliferation or their proliferative response. And what we see, surprisingly, and these are pictures of those wounds, and so each one of these black marks is a new neogenic hair follicle. We see a huge increase in the number of new follicles that are generated within these wounds when we knock out or temporarily knock out HIC1 during this wound healing process. And so this is the quantification of that on the left. So now we're seeing around 35 new follicles that are being generated within those wounds. So it's almost a sevenfold increase in the number of follicles that are being generated. And so this tells us that in fact increased numbers of fibroblasts within a wound is not necessarily a primary contributor of fibrosis or hyperproliferative scarring, but it's actually also the wound environment itself that may be determining whether that wound is fibrotic or regenerative. To summarize, what I've told you so far is that the progeny of HIC1 lineage progenitors are a primary source of wound healing fibroblasts. Different wound microenvironments drive divergent transcriptional programs, and these promote acquisition of fibrotic or regenerative fates. And this is achieved by both activating unique gene expression programs that often are reminiscent of skin development, but they also modify chromatin accessibility to key transcription factors that we had identified. So our ongoing work is actively investigating the extracellular factors that comprise this pro-regenerative microenvironment, and we were keenly interested in the immune response within these wounds. And I'll touch a little bit on that in this next section of the talk. So in doing this work, one of the things that we began to appreciate is the importance of the model that you used in order to study wound healing. And there are a number of different models that are used to study tissue regeneration. One, for example, a powerful model is the axolotl that's used for limb regeneration. In mice or in wound healing, another new model that's come around is the African spiny mouse, which has an enhanced ability for regeneration following wounding. But mice in general are actually quite a poor model of human skin wound healing, particularly because mice are loose-skinned, and they have this structure called the panniculus carnosum muscle layer in the deep layers of their skin that allows them to heal wounds primarily by contraction. And humans don't do that. So humans actually heal wounds by cell proliferation, migration, and production of extracellular matrix. And so translating things that we see in mice is often, I think, will be quite difficult to to predict what will be effective in humans based on these very different mechanisms of wound healing. The other thing about laboratory mice is that they lack genetic diversity, and they have a relatively uneducated, given that we raise them in very sterile living conditions and lack sort of a wild microbiome. And so what we tried to do was to think of models that could overcome some of these obstacles that might better reflect the wound healing process that occurs in humans, but also might offer some new insights into adult mammalian skin regeneration. And so I'm going to tell you about a model that I'm sure you've probably never thought about or never thought you would hear about in the context of skin wound healing, and that is reindeer. And so in Calgary, we have, as you heard at the beginning, you heard that I'm part of the Faculty of Veterinary Medicine. And as part of our faculty, we have a herd of caribou or reindeer that has traditionally been used to study climate change and the impact of climate change on parasite behavior and on vulnerable species like reindeer. But for us, the interest in this species was their ability to regenerate their antlers. And I'll tell you why. The deer are really the only mammals that can regenerate entire organs of any mammal. Reindeer antlers are regenerated every year in both males and females, which makes them unique from other deer species. And I think what is most remarkable is that these antlers grow at a rate of 1 centimeter a day. And so what that means is that you're actually generating almost 12 centimeters cubed of tissue every single day as they grow their antlers every spring. The other interesting thing is that that reindeer can actually regrow new antler branches if a branch is amputated or injured. And that the antlers are covered in this hair-bearing skin called velvet. And it's not just simply hair-bearing, but these are this is in every way a complex piece of skin, much like is the human skin. It has a variety of sebaceous glands. It's highly innervated, a massive vascular supply. And this is a tight-skinned mammal. And so I think this offers a number of advantages and a unique opportunity to potentially study this velvet as a model of tissue regeneration. And based on the ability of this animal to regrow new antlers, we hypothesized that this velvet that covers the antler might also harbor some enhanced regenerative capacity that could provide a unique model to study skin regeneration. So to test this, we started to do wound experiments in these animals. And so what we did was we made 12 millimeter wounds, basically full thickness biopsy punches through the antler velvet, which is so the antler velvet, or we made identical wounds on the back skin of that same animal. So now we have a comparative model where we can make 2 identical wounds in 2 different sites and then compare the outcomes. Okay, so here's what this is on day 0. This is the initial wound on the back skin, and then 30 days later, this is what it looks like. And so you can see you get this sort of slightly raised, irregular scar that occurs. Similarly, we made burn wounds in order to study the response to a burn injury. So here's the burn wound. This is the same size as the excision wound, and 30 days later, now you see this very nice scar, much like you would see in humans. In contrast, here's what we see when we make those identical wounds in velvet. So this is what it looks like on day 0. 30 days later, you'll see that this has started to repigment, and then 60 days later, you can see that this is completely regenerated with new hair follicles that are covering almost the entire surface of that wound. Similarly, after the burn wound, so here's what the burn looks like. This is at 30 days. You can see this is starting to repigment, unlike in the back skin where you get very little repigmentation. You can see that pigment is starting to come from the out. This small, but by 60 days, again, you can hardly see where that initial insult took place. And just for to assure you that these burn wounds are full thickness, you can see that this is 3 days after one of these thermal injuries, and this is now looking from the ventral surface of the wound or of the velvet. You can see that this is all the way through. You've got full thickness damage to that velvet, and yet it is still able to clear all of that debris and fully regenerate that skin. So here are cross sections of what the skin looks like at the 30-day time point following injury. So this is the back skin wound. So you see this hypertrophic epidermal layer here, which is thickened, but with the arrows mark the margins of the scar, and you can see that you've got this sort of raised scar, but you see a complete absence of follicles within that back skin wound. And this is just a closer view of that here. In contrast, what you see in the velvet is you can see that you've got regeneration of follicles almost throughout the entire extent of that wound at varying stages of development. I should point out that they regenerate from an outside-in fashion. And so you've got more mature newly formed follicles on the outside, and as you move towards the inside, you've got more immature follicles. But you can see the difference at high magnification, where these follicles, you can see at the bottom, these are proliferative epithelial cells at the bottom of each of these follicles, and you see an absence of that in these sort of ridges, epidermal ridges that are formed that never actually go on to form follicles. This is just some bright field images showing the absence of repigmentation. This is these two examples are from burns. So you've got an absence of repigmentation and an absence of new follicles in the back skin injuries. But 30 days post burn, you can see that you've got near complete or scarless healing that occurs within the velvet, and these beautiful follicles that are forming. And this is just that higher magnification view where you can see the pigmentation and these new follicles that are forming, along with various glandular structures that are formed in there as well. And the quantification at the top just shows you that that in back skin you're getting less than 20% of the return of hair follicle density within those wounds. It's probably less than that actually, because many of the follicles that do form are irregular and likely dysfunctional. But in the velvet, what you see is around 80% of return of hair follicle density within that wound. So you're getting a substantial amount of regeneration. So the next question that we asked was whether this whether the regenerative response is due to their location in the velvet or in the antler, or whether this is due to essentially whether this is due to some factor within the antler, or is this regenerative competence inherent to the velvet fibroblasts themselves? So we did a transplant study where we took velvet, full thickness velvet, and grafted it to this non-regenerative site, which is on the back skin. We waited for those grafts to heal, and then we made a full thickness wound within that graft to ask whether it was still able to regenerate. So here's an example of what one of these grafts looks like, a cross section of the graft. So this is what the graft looks like on the back skin. So you can see the difference between the hair types between the graft and the wound. This is showing you just a cross section. This is the back skin. You can see these large insulating follicles versus the smaller follicles that are in the velvet. And then this is the result. So we've marked the margins of the wound at the time of injury using red fluorescent beads. So this is the two outside perimeters of the wound, and you can see that those are that the inside of that wound is completely filled in, and you can see that there are in fact new hair follicles throughout the width of that injury, and again you've got a no difference in terms of the density of follicles within the wound versus the outer area. So what this tells us is that regenerative competence is actually inherent to the cells within the dermis and is not due to their location or some indirect effect of being located on the antler itself. So what distinguishes the velvet fibroblast from the back skin to enable regeneration? So to answer this, we again undertook single cell RNA sequencing, and we profiled all of the cells within the back skin or in the velvet, either before or after injury. And what was most striking was that the population that showed the biggest difference, and we did this in an unbiased way, looking at perturbation scores, was that it's actually only the fibroblasts that actually differ. So you can see that this is the back skin fibroblasts here, and then this is the velvet fibroblasts here. You can see that there's a complete absence of this of some of these fibroblasts within the velvet. And so this suggests that there is a very different fibroblast transcriptional profile relative to those that are found in the back skin. And so then we compared transcriptional profiles between the velvet and the back skin, but we also compared these to human skin, derived from, so human skin fibroblasts derived from either fetal skin or from adult healthy skin. And this is taken from data derived from this recent paper by Reynolds et al. That was published in Science. And so this is a consensus plot showing these genes that are enriched within the regenerative velvet against genes that are enriched within the back skin or fibrotic region, and then they're overlaid with and they're compared to genes that are enriched in either regenerative fetal skin or non-regenerative human adult skin. And what you see is that there's almost a complete overlap and conservation of regenerative programs that are observed between fetal and velvet skin, and similarly between back skin, reindeer back skin, and adult human skin. And so what this tells us is that there are discrete programs that are driving these very divergent fibroblast behaviors. And you can see just with these violin plots, a sampling of some of these genes. So something like midkine is highly expressed in the fetal fibroblasts and in the velvet fibroblasts. Same with CRAB-BP1. And then you get these more inflammatory or pro-fibrotic genes that are enriched within the reindeer back skin, as well as in the back skin, which is in green, and in the adult human fibroblasts. Interestingly, many of the same genes that we identified as being pro-regenerative or pro-fibrotic in our mouse models are also showing up in the reindeer velvet model and back skin model. And so things like HDAC2 and LEF1. So this is looking at scenic analysis. We can see that the velvet cells, which are primarily on the left side of these plots, are highly enriched for network activity related to HDAC2, as well as LEF1 signaling, which is a key regulator of Wnt signaling. But then it's those the back skin fibroblasts that are enriched for inflammatory transcriptional programs like NF-kappa B1. So if we look at the trajectories of these different fibroblasts over time after the injury, we see a very unique phenomenon happening. So this is now looking at SC-VELO, which shows vector fields, which is basically infers. So it basically takes advantage of ratios of spliced versus unspliced mRNA and unspliced increases in unspliced mRNA typically indicates upregulation of a gene. But you can use these ratios to basically predict future states of fibroblasts in this case. And so when we look at the profiles of fibroblasts over time in response to injury, the back fibroblasts, you see that
start off in
the top regions of these plots, they start to shift downwards, and they basically end up in a completely different space. And so what this suggests is that these are adopting an entirely new fibroblast phenotype in response to the injury, and this is likely representative of this fibrotic phenotype. Interesting, what we see in the velvet fibroblasts is again this shift downwards to and almost an overlapping phenotype at day 7 with what we see at back skin. But we see this reversion that occurs between day 7 and day 14 back to their original ground state phenotype. And you can visualize that a little bit better here when we look only at the day 7 to 14 profiles, where we're seeing in the back skin, which is indicated by these blue arrows, the trajectory is actually moving downwards, whereas in the velvet, which is in this pink arrow, is actually moving backwards, back up towards their original phenotype, which is here. And so we're seeing this very divergent response that appears to be driven by a reversion to their original phenotype. And so to look at what might be driving this divergence, we looked at genes that are particularly active during that, or preferentially active in either back skin or velvet during that 7 to 14 day transition period. And again, we see many of these pro-inflammatory genes that are being activated in back skin, so IL-6, SAA3, CCL5, and IL-1A, all being increased when you plot these on pseudotime during those late stages between days 7 and 14. Whereas in the velvet, we see enrichment for genes that are associated with acquisition of inductive phenotypes in mesenchyme, and also in immunosuppression. So things like CCL26 and CXCL14 also going up in these cells. So it's the onset or enabling of these regenerative programs to be engaged at that 7-day time point that's allowing regeneration to proceed. The other thing that we looked at was how, given the fact that we see all of these inflammatory genes being activated within the fibroblasts, particularly at this day 0 time point, we thought that maybe these fibroblasts might be initiating an inflammatory priming that might modulate or alter the immune response to this tissue injury. And so what we did was to basically look at all of the immune cells over the course of this wound response in either back skin wounds or in velvet wounds. And this is a bit of a busy slide, so I just want to just highlight a couple of things. At day 0, there's actually, to our surprise, a greater concentration of immune cells within the velvet than there is in the back skin at rest. But what's remarkable is the difference in the immune response at day 3, in that the back skin initiates a highly intensified macrophage response. So these CD68 positive, NOS positive, and DUSP2 positive cells are highly enriched relative to what you see in the velvet response. So you're getting a very different immune response that's being elicited in response to the injury between these two sites. At day 7, you start to see this starts to catch up in the velvet, where you're getting a more robust macrophage response. But interestingly, this resolves almost entirely by day 14, whereas the immune infiltration persists in the back skin. And so you've got very different immune dynamics occurring within these two sites. And we think that that's potentially feeding back onto the fibroblasts at these later stages of wound healing, and maybe, in the back skin, actually suppressing the regenerative response that might normally occur. And so the last thing I want to tell you before I end off is now probing this crosstalk between fibroblasts and immune cells. And so what we did was to really focus in on day 0, this ground state priming event, and look at the factors that are being produced by fibroblasts to potentially influence this initial immune response to injury. And so these are Circos plots, where in red in the bottom, these are all the ligands that are being generated by fibroblasts in velvet. And then what we do is to look at all of the other cells within the velvet that might express receptors for those particular ligands. And what you'll notice is that all of the receptors and the cell types that are potential receivers of those signals are typically vascular smooth muscle or endothelial cells within the velvet. Now, in contrast, if you look at the back skin, you see that the fibroblasts are secreting a very different set of ligands, and the receivers for those ligands are also very different. And the largest receiver for those is actually macrophages. And so this really tells us that that pre-injury communication is already biased towards priming macrophages even before the injury starts. And so and probably priming them to be in a hyperinflammatory state. And this may, we think that this might actually be one mechanism by which we are biasing the immune response towards a fibrotic response in adult mammals. And so the last thing is that we tested this by basically taking these factors that are being produced, a series of these factors that are being produced within the back skin and not in the antler velvet. And we took these factors and then applied them to the antler velvet at the time of injury, with the intent of asking whether we could block regeneration by providing some of these priming cues in order to potentially alter the immune environment or that skin milieu to see if that was sufficient to basically block this inherent regenerative process that occurs in regeneration. And I'm not going to get into the specifics of these, but these are 3 examples of where we've applied some of these either inhibitors or activators of these different signaling pathways that are enriched in the back, applying them onto the velvet. And in every case, what we can see is a near complete blockade of regeneration and initiation of what looks like hypertrophic scarring that we might see in the back skin. So you can see, the black dashed lines indicate the margins of the wound, but you can see that there is almost now a complete absence of hair follicles within the wounds. In some cases, we get this sort of hyperplastic epidermis. We see these elongations, very much like what we see in the back skin wound healing process, but we never see these actually emerging to become hair follicles, which is exactly what we see in these back skin wounds. And then in some cases, we're actually seeing these very robust scars forming on the velvet, which we never see in vehicle-treated or controlled empty wounds. And so just to finish off, this is our current model. We think that unique transcriptional programs are engaged in wound responsive fibroblasts, and these are active in both uninjured and injured velvet and closely resemble regeneration competent programs that are previously identified in mice. Dermal fibroblasts are key orchestrators of the immune response to injury, and that these interactions are a critical determinant of subsequent wound healing events, including fibroblast fate. And in postnatal mammals, dermal fibroblasts adopt a state of heightened resting inflammation or priming that we think incites an amplified immune response that promotes more rapid wound closure and repair at the expense of regeneration. And so the way we think about this is that the dermal fibroblasts in the velvet secrete a certain set of factors that allow the immune response to occur, but immune response is permissive for regeneration to occur. But in the back skin, this priming elicits a different set of factors that then amplifies the immune response, that then blocks or suppresses the natural regenerative response and drives these fibroblasts into a fibrotic state. And so what we're hoping to do is to capitalize on this information to start to develop potential antifibrotics that one of my students has coined as being stromal immune crosstalk decouplers that we hope might actually mitigate some of the scar that we see in patients. So I just want to acknowledge the people that have done this work. So Holly Sparks and Sartak Sinha really led all of the work that I told you about in reindeer, along with Arzina Jaffer, who did some of the bioinformatics with us. Haley Robbins was also instrumental in this work, as well as our collaborators on the large animal side, Dr. Rob McCorkle and John Mattias. And I just want to acknowledge CIHR and NSERC funding, who supported this work, as well as our longtime supporters, the Calgary Firefighters Burn Treatment Society, who sponsor a lot of this work. I'll put a plug in to say if anyone is interested in working on fibrosis and regeneration in skin, we're currently recruiting at the doctoral and the postdoctoral level, and I'd be happy to take any questions or discuss any of this work later on. So I'll stop there. Thanks very much for your time. And I apologize for over.
No, that was a very, very exciting talk. I think especially the reindeer stuff was quite a treat for a lot of us. It's a really new model for our students and staff to see. So now we'll just open up the floor for the questioning period. So you can just go to the Q&A session on the side and ask any questions that you might have and vote on them, and we'll read them out for everyone. So while we give people some time to do that, I just had a question on behalf of kind of the student leads. So we were wondering, for your reindeer models, what is it like in terms of reindeer age and maybe the seasons in which you're doing these experiments? So our assumption is kind of that older reindeer might have less regenerative abilities, or seasonal differences might affect the growth rates of the antlers. So with this, I assume it's hard to control kind of these variables.
Yeah, you're right. So yeah, there's a couple things that you should know. So the velvet is only, every spring, so sometime around March or April, every antler or every reindeer will generate new antlers, and in that process is when they have that velvet that covers the antler, and then by about late summer or early fall, all of that velvet is sloughed off. So it basically degenerates and is sloughed off, and then you're just left with hard antlers, so that bony antler that you typically see. So we've got about 4 months to actually work on these velvet wound healing experiments. So we, in answer to the first question, which is age, we have been doing, so the animals regenerate antlers even in their first year of life, and then continue to do so throughout their lives. And with every year, those antlers get larger and larger. There's really no difference in the ability or the velvet to regenerate as a function of age. So the older animals are just as likely and just as effective at regenerating those wounds as the younger animals are. The only place where we see differences in rate of regeneration is if you make a wound at the tip of the growing antler versus at the base of the growing antler. And this is really just based on most likely the number of actively dividing cells that may be present within the velvet itself. But it's not that it doesn't actually regenerate, because in both sites it will regenerate to the same degree, but it just takes a bit longer to regenerate at when you make wounds at the sort of the base of the antler as compared to the more distal regions of the antler itself. And this may also have something to do with changes in blood flow at these different sites as well. But certainly that's the only place where we see real differences.
Okay, and so in the chat, our director Ming Yao has a question, or two questions, but one of them is that it seems like in humans, scars can reoccur after some sort of surgical removal or steroid injections, and it seems like there might be an element of genetic inheritance in this effect. So he's wondering if you have any insights on this.
Yeah, I'm sure that, I'm sure that there are genetic, like heritable impacts on wound healing and scar formation. I mean, a keloid would be a good example of that, where you get this exaggerated scar forming process, and this can be heritable. And so, I certainly think that there is a component to that, and I think that mutations that might affect immune responses, as well as mutations that might affect fibroblast behavior, could all impact wound healing. And what we're starting to appreciate is that it's this ensemble of players that all sort of, and it's that interplay between those different cell types that ultimately influence the outcome of the scar or the regeneration of that wound. And so I think any perturbation to various cell types could influence that outcome. So, and I don't know about steroid injection. That's a good question. I don't know about how that might influence things.
Okay, and so for the next question, it looks like we'll have invite someone to share with their audio. So here we go.
Hello. A bit long out of the state via microphone. But it's all fascinating talking to you very much. I don't have much background in this. I just want to increase my understanding. From what I understood of your talk, there's essentially two ways to translate into the clinic how you would fix the regeneration without scarring. And from what I understand, sort of mimic the more regenerative site. In my case, be the middle region, or in reindeer, it's being the velvet, and you would provide the environment, of immune environment that mimic that. And what I understood was that you would sort of get a graft of that better regenerative. So I was thinking, when it comes to humans, which better? And you think the better regeneration is better? How would you go about to modulate all of the genes and the different epigenetic factors and such? And if you think that a graft is better, where would be the source of a better regenerative site in humans? Maybe, as was said, from the base. Thank you.
Yeah, that's a good question. So, yeah, sorry if I wasn't clear about this. So, I don't think that grafting anything, although I guess you could graft cells. I think certainly transplanting cells that may have a greater competence, supplementing them into, say, skin grafts, for example, which is something that we have a lot of interest in. We've done that, supplementing dermal progenitors that we've isolated from skin and from hair follicles, and put those into split thickness skin grafts in order to regenerate a more functional dermal tissue beneath the graft. And that has shown some functional benefit actually in graft function when we look at the biomechanics of the graft, as well as itch and pain responses. And this is all in animal work, but also in mitigating some of the scar or the collagen, the aberrant collagen production within the graft itself. And so I think there is a place for potentially grafting cells that make more regenerative into say split thick to improve. But I don't think there's I wouldn't say that grafting whole pieces of skin will likely change things. But I think if we're moving into the clinic, what I think our data is telling us is that there are really 3 mechanisms that we need to attack in order to mitigate scar. The first one is that you have to have a fibroblast that has a transcriptional program that is competent to initiate regeneration, that is competent to talk to epithelial cells in the epidermis in order to re-engage those developmental processes to produce new appendages, right, like hair follicles and glands and things. But also fibroblasts that are engaging those processes and are not producing excessive amounts of extracellular matrix, right? And part of that is that immune response piece that I also told you about. This is the second really key avenue that I think we can attack in the clinic is by modulating the immune response during those very acute stages of wound healing. And so everything we've done, and also in experiments that we've done that I haven't told you about, we know that at least in mouse models, it's that first week after the injury that is really critical in determining the outcome of that wound, whether it's regenerative or fibrotic. And so you can, things that you do following that 7-day period have very little impact on regeneration. It's almost too late to salvage. So you need to be changing the immune response very early on during that acute period in order to make that environment permissive for fibroblasts that are regenerative to actually engage those processes. Okay, so I think we have to make a permissive environment within the wound, and that means modulating the immune response. So changing the types of immune cells that are being recruited, and then the quality of those immune cells. So it's not just suppressing all macrophages, but actually engaging certain types of macrophages or certain types of neutrophils in order to that are better able to support those regenerative functions of the fibroblasts that are being recruited in. And then the last thing, whether any of these things are more important than the other, I think those are probably the 2 most important things. The epigenetic changes that we observe may be a result of the inflammatory influence of the immune system on the fibroblast, right? And so by disengaging or modulating the immune response, then you may enable these epigenetic changes to occur or not occur and make this more permissive for regeneration. So I think for me, it's really a 2-pronged approach, which would be modulating the immune system and ensuring that you have fibroblasts there that are able to engage regenerative programs. And that might also involve some sort of in vivo, like in situ reprogramming of fibroblasts. So some direct reprogramming process where you purposefully activate certain transcription factors like CRAB-BP1 that we've identified, but there are likely other ones like LEF1 that other labs, Ryan Driscoll's lab has identified, other ones that might be key for the regenerative process.
Thank you very much.
You're welcome.
All right, and then we'll have time just for one more question. So Daria is asking in the Q&A. So firstly, what a model. I assume she means for the reindeer. It's very exciting. Have you tried to prime with these velvet factors, the wound on the back, to see if regeneration occurs? And similarly, applying this to your experiments where you're stopping regeneration with the velvet.
Yeah, so that's exactly what we're trying to do now. So we're doing both of those things over this summer, and that is, continuing on with identifying factors that block regeneration to identify those key processes, the processes that are most important for enabling regeneration in the velvet. But yeah, then taking factors that are unique to the velvet and trying to apply that in the back. And this is a bit more of a difficult task, because like I said, I think you have to have both of these processes occurring at the same time, right? So you've got to have modulation of the immune system, and I think it's likely multifactorial. It's not going to be just a single factor. We've tried a couple of these things in our mouse models, and like as I showed you, even initiating or application of single factors, you get a little bit of a bump in regeneration, but it's still pretty modest, right? And so I think this is going to be a combinatorial approach with multiple factors that need to be applied, and so we're trying to work through some of those permutations now, identifying which ones are most important, and then applying them simultaneously to those early stages during wound healing. Yeah, that's a great question. So that'll be over the next 5 years.
Awesome. So with that, I would like to take this chance to again thank Dr. Biernaskie for joining us today. It's a virtual round of applause on behalf of everyone. So right now we'll take a slightly shortened lunch break. And so we'll have an opportunity in one of the networking sessions for people to just join in and have lunch for us with Dr. Biernaskie, but I guess maybe breakfast for him. And you can ask him any follow-up questions about research or career or anything like that, so join us there. For new students, there's actually a Q&A session, and that's hosted by our admissions officer, Hazel Pollard, and one of our PhD students, Michelle Dubinsky. And then we'd also like to remind people that in the Expo tab, you can find a bunch of booths from some IMS affiliated groups, and those teams put a lot of effort into those, and they're absolutely wonderful, so please take a look there. And then finally, just a quick reminder that Data Blitz will begin at 1, so join us for some more student talks in neuroscience, clinical research, and in basic sciences. And with that, we're ready to go to the next steps. Thank you again.